my pleasure to introduce today's first speaker, Dr. Tyler Doyon, who I had the pleasure of working with in my group as a PhD student. But before he came to Michigan, he studied at U of M Flint, on the campus in Flint, where he majored in biochemistry and had the opportunity to get some inorganic research experience. And upon joining the program in chemical biology at Michigan for his PhD, he joined my group, which at that time had 0 members. He was one of the first students to join the group and really helped build all of our research programs. And Tyler made a big impact in a lot of areas, but he spent the majority of his PhD focusing on alpha-ketoglutarate dependent non-heme iron enzymes. And so that's what we're going to hear from him today. And I'll mention that he did recently defend, so he's Dr. Doyon now. He defended in the beginning of September 2020 and has moved on to a postdoc position in Andrew Buller's lab at Wisconsin. And we got a taste of the research going on there during the April meeting. So
good morning or good afternoon, depending on what side of the Atlantic you're on. Very happy to be here today and to talk to you about some of my research from my time at Michigan, and particularly a story about the mechanism of tropolone biosynthesis as catalyzed by a non-heme iron dependent dioxygenase. And so, as we've heard many times in this particular forum, we've talked a lot about non-alpha-ketoglutarate dependent non-heme iron enzymes, which are these really super biocatalysts which catalyze a variety of oxidative transformations and couple their the decarboxylation of alpha-ketoglutarate and other alpha-keto acids to the formation of these iron 4 oxo species, which are capable of a wide variety of highly selective oxidative chemistries, particularly in context of natural product biosynthesis. And so with a few examples, selective epoxidation reactions can be catalyzed by these enzymes, endoperoxidations, radical cyclizations and ring rearrangements, ring contractions, hydroxylations, halogenations, and ring expansions, just to name a few, are all catalyzed by this one particular class of enzyme. And so we're very interested, obviously, in studying this class for its synthetic potential, and particularly its synthetic potential for generating tropolones, which are this really unique class of natural products which possess a wide variety of biological activities and are primarily constructed of this tropolone core, which is the 7-membered aromatic alpha hydroxy ketone core. And of course, this core can be decorated with a wide variety of functional groups, including some crazy meroterpenoids and some other interesting functionalizations. But the activity of the tropolone is actually thought to primarily stem from its the ability of the tropolone moiety to bind divalent metal ions. And so this class is of particular interest to both synthetic chemists and medicinal chemists as potential targets, potential inhibitory target molecules. And so we're particularly interested in ways that we can quickly access this core, and in particular, the way that nature is able to access this core so efficiently. And sort of the seminal work in this area really comes from Russell Cox and coworkers back in 2012, when they identified the biosynthetic pathway of stipitate acid in the fungi Talaromyces stipitatus. And this particular natural product is known to arise through really this 2-enzyme cascade. First enzyme is a flavin-dependent monooxygenase, which catalyzes the oxidative dearomatization of this 3-methylorcinaldehyde substrate, generating this dienone, which is the substrate for Trop C catalyzed ring expansion. And Trop C is a alpha-ketoglutarate dependent non-heme iron enzyme. And so this natural product is actually decorated further downstream in downstream reactions with additional enzymes to generate stipitate acid. And in recent years, there have been additional tropolone containing natural products which possess this stipendaldehyde core, particularly a lot of tropolone meroterpenoids, whose biosyntheses have been examined and found to proceed through the same two enzyme catalyzed pathway. This flavin-dependent monooxygenase dearomatization followed by a non-heme iron catalyzed ring expansion to generate the tropolone core. So we were very interested in the way that this non-heme iron enzyme is able to catalyze this ring expansion. It's a unique reaction amongst the non-heme iron class of enzymes. And so we wanted to really study the way that this is happening. And we noted, as well as Russell Cox's group noted, the formation of stipendaldehyde in reactions with wild-type Trop-C, as well as this shunt product. And originally it was proposed that this particular transformation could occur through an initial H atom abstraction to generate a primary substrate radical, which would then undergo sort of the canonical rebound hydroxylation as catalyzed by non-heme iron enzymes to produce this 1,2-diol. And this dearomatized diol was proposed to undergo then a semi-pinacol rearrangement, presumably catalyzed by local acids and bases in the active site of the enzyme to ring expand and produce the aromatic tropolone. Alternatively, this diol could undergo a retroaldol fragmentation and re-aromatize to form this 3-hydroxyorcinaldehyde shunt product, as is observed in small amounts in wild-type reactions. So we're particularly interested in the reactivity of this diol, as the idea that it could be used as a way to generate tropolones was particularly appealing from a synthetic standpoint. And so we investigated the reactivity by coming up with sort of a workaround for accessing this molecule without involving the non-heme iron enzyme Trop-C. Instead, we used a non-heme iron enzyme which performs selective benzylic hydroxylation to generate this benzylic alcohol, which we then dearomatize using the canonical pathway Trop-B flavin-dependent monooxygenase to produce the diol directly. And what we didn't know at the time is whether this would ring expand to form a tropolone or would instead give us some other product. And it turns out that it does only deliver the shunt product in reactions to generate this diol. And in fact, that reactivity is so fast that we never actually observe this diol in solution. Additionally, so at this point, we thought we would maybe be able to trap the intermediate by the addition of Trop-C to the reaction mixture. And the idea here was that we would generate the reactive intermediate in situ, which would then presumably diffuse into the enzyme and undergo the ring expansion chemistry as had been previously described. And instead, we actually observed complete conversion again to this shunt product. We never observed any tropolone in reactions with this diol intermediate. And that really kind of made us think about the mechanism of this, or particularly the reactivity of this intermediate as it relates to the tropolone ring expansion reaction. So it seems that outside of the enzyme, we are seeing only the shunt fragmentation pathway to deliver this shunt product, whereas inside the enzyme, we're clearly delivering the ring expanded product. So there must be a very particular set of acids and bases or polar residues in the active site which are catalyzing this transformation that are key to the ring expansion activity that we observe in Trop-C. So we really needed more information about our enzyme. And so we teamed up with some really great structural biologists, Dr. Lena Malik and Professor Marcos Koutmos, who were able to help us solve a strict crystal structure of our enzyme. And we were able to finally visualize the active site in a meaningful way. In addition, we teamed up with a team of computational chemists, Kevin Skinner, Dr. Troy Wymore, and Professor Paul Zimmerman, also at the University of Michigan, who were able to model in, using MD simulations, our substrate and dock that into the active site of our crystal structure. And so we could see the dearomatized substrate in a catalytically active position with its methyl group pointed toward the non-heme iron center, as well as the interactions with the ketone groups of our substrate with several local residues. And so with this information, we also were able to access basically give us a reference point for a bunch of residues that we were interested in mutating to probe this mechanism. And so we went pretty liberal in choosing our residues because we weren't sure exactly which residues could be involved in the mechanism. But I went forward with this mutagenesis. And the idea here was that we would probe the ring expansion activity, which with wild type Trop-C delivers very consistently a high amount of stipendaldehyde and a very small amount of this shunt product. We envisioned that adding a Trop-C variant, which perturbs the ring expansion activity of the enzyme, would enable us to see a huge drop in the amount of stipendaldehyde produced and a subsequent increase in the amount of shunt product that is generated. And so looking at all of the polar residues that we thought could be involved in the ring expansion reaction and doing an alanine screen on all of these residues, we found a few that were catalytically inactive, which is maybe not surprising. And in the case of those that were catalytically active, we saw mostly wild type activity. So maybe not to the extent or the conversion that we once had, but you can see in all these cases that stipendaldehyde is still the major product that's generated in this reaction. And we would have expected this shunt product to be the primary product if we were truly shutting down the ring expansion activity. And it was at this point we started to maybe sort of question the identity of this diol as the correct ring expansion intermediate. And we had looked at some other literature surrounding this area going back to 1984 when the Reese group attempted to make tropolones through a ring expansion mechanism that's was presumably going to be very similar to what is proposed for our enzyme. And so by adding nucleophiles to these dearomatized spiroepoxides, they attempted to perform a ring expansion, but instead only saw this similar type of shunt reactivity, this retroaldol fragmentation activity. And they explain this as being primarily driven by the energy of rearomatization, and they did not observe any ring expansion products in these efforts. Now, very recently, David Sarla's group released a dissertation where they examined a wide, diverse number of conditions, expanding on what the Reese group had explored, trying to do the same reaction, trying to make a tropolone through a semi-pinacol rearrangement. And instead, all of their conditions also showed retroaldol fragmentation as the only mode of reactivity for that for the intermediate that's generated, the diol intermediate. And so based on that, the comparison to our own intermediate, we can see that there are obvious parallels here with this diol intermediate quickly undergoing this fragmentation reaction in all of our reactions to generate that intermediate. And we began to think about other possible mechanisms by which this ring expansion could actually occur that might not go through the diol intermediate. And so it's well known with non-heme iron enzymes that they can also pursue radical mechanisms and can actually achieve a lot of ring modifying reactions through well-known radical mechanisms. And so we then propose that, or hypothesize really, that this could we could generate a primary substrate radical, which would then quickly rearrange to form a tertiary radical, and then be quenched by the iron 3 hydroxyl species to generate stipendaldehyde as the product. In the alternative mechanistic view, you could undergo rebound hydroxylation, which we know is the primary source of our shunt product based on our reactions. And so we hypothesized that this could be the sort of the diversification point for whichever product that you actually observe in these reactions. And it turns out that this radical mechanism is actually precedented in the synthetic literature as well. The Topke group back in 1987 proposed a radical or generated tropolones through generation of a pseudobenzylic dearomatized radical species, which then rearranged to generate these protected tropolones. And so if you think about our mechanism, it's completely analogous to what they've actually directly done here in generating tropolones through production of this initial radical species. If we also consider the differences in the model here, so this is a rather simplified version, but if we think about the especially from the perspective of non-heme iron halogenases, the rebound hydroxylation we know is preferred when the substrate alignment relative to the iron 3 hydroxyl species is good and in close proximity. Enzymes which non-heme iron enzymes which proceed through alternative mechanisms other than rebound hydroxylation typically have poor alignment of the iron 3 hydroxyl to their substrate, which enables this other activity to be the primary mode of reactivity observed. And so we began to think about the way that substrate positioning could impact the distribution of products that we observe in this reaction. So again, going back to our MD model and looking at a couple of aliphatic residues, there's a couple of additional ones surrounding this that we observed as well, but in particular, these two phenylalanine rings, which flank our substrate in the model and could potentially, direct whether the distance from the substrate radical, which would directly result from CH atom abstraction, to the resulting iron 3 center. And so mutating those residues, we again did an alanine screen and showed that the F284A, which I'll show again in a moment, generates, basically this variant generates nearly equal amounts of trihydroxybenzaldehyde, the shunt product, and stipendaldehyde, the tropolone, which is suggestive that we've now modified the way that that substrate is sitting relative to the iron 3 center. And if we restore, if we mutate this to a tyrosine, which is isosteric and isoelectronic to phenylalanine, I think we definitely see a restoration of nearly wild type activity, a little bit more shunt product formation, but nearly wild type activity in those assays. And so this really suggests that the substrate positioning could play a key role in determining what products we get out, and in fact does. So the final piece that we wanted to investigate, and again, teaming up with our computational collaborators to do this, is to look at the thermodynamics of these transformations and kind of get a sense of what the barriers are for these reactions. And so to do this, we actually teamed up with, again, Kevin Skinner and Professor Paul Zimmerman to construct these pathways. And so if we look at the radical rearrangement first, starting with our primary substrate radical at the 0 point energy, it's about a 12.7 kcal per mole barrier to undergo this radical rearrangement. And in this case, it is a pretty reasonable barrier for that reaction to happen. If we start with our diol, which is obviously starting at a much lower energy point than our free radical, we see it's about a 50 kcal per mole barrier to undergo this semi-pinacol rearrangement, which is in line with our other observations from these reactions. And similarly, if we look at the fragmentation pathway to generate the shunt product, we see that retroaldyl fragmentation is in fact a low barrier process of about 18 kcals per mole, which is again in agreement with what we had seen previously. And so to conclude, we found that the radical rearrangement is a low barrier process, which very likely is generating tropolone in these in the context of these particular non-heme iron enzymes. The rebound hydroxylation appears from all of our data to only generate 3-hydroxyursinaldehyde, the shunt product, and appears to be the source of shunt product formation in the wild type reactions. And we know that this is more favorable than the ring expansion reactions by about 30 kcals per mole. So with that, I'd love to thank the team in the Narayan Lab, especially Allison, for fantastic mentorship over the years. Team non-heme, that would be Jonathan Perkins, Evan Romero, Kendrick Smith, Dee Yang, and Summer Baker Dockery, as well as our computational collaborators, Professor Paul Zimmerman, Troy Wymore, and Kevin Skinner, and our crystallography gurus, Marcus Kumos and Lena Malik. And I'd like to thank you all for your time today, and I'd be happy to answer any questions that you might have.
Thank you so much, Taylor, for your exciting talk about ring expansion. It's great to see all the enzymes that help us to make new aromatic molecules. So the talk is now open for questions. Anthony?
Hi, Tyler. We have someone with their hand raised in the chat, so I'm going to try and allow them to talk.
Okay.
Let's see if this works. So it's Hai Yang. Would you like to ask your question?
Maybe not.
Maybe it's accidental, Tyler. Maybe I can go first. Is it maybe a couple of questions? Like, so one of them more experimental. So is it possible to deuterate the methyl group in those molecules? And have a look at some kinetic isotope effects there, which obviously will give you some strong evidence for
that initial. Sure. Yeah, it's definitely something that we've thought about. It wouldn't necessarily tell you, of course, the divergence point, because they would both, the H atom abstraction would be the initial step in both mechanisms, but it's absolutely possible that, yeah, doing that initial, demonstrating that the initial H atom abstraction happens on the methyl group would be a great way. We just hadn't pursued that.
Yeah, that's right. And then maybe as a follow-up there, so like you talked about substrate positioning in these enzymes, which clearly is very important. But of course, the substrate has to be positioned for the CH abstraction still. And so what kind of strategies are we talking about here for allowing CH abstraction to take place, but not allowing the rebound to take place?
Yeah, so it seems like in the case of most of these non-heme iron enzymes that, especially like halogenases, that the active site is very constrained, where the initial H atom abstraction can happen, but there is some sort of conformational change or some sort of distance. I guess, a constraint that sort of pulls back on the substrate, and also there are potentials for potentially some isomerization of the hydroxyl group in the active site as well. So it's not really known in this case how that would happen. We're pursuing some QMMM calculations to sort of parse that and see if we can understand a little bit more about our variants that we've generated as well, how that might happen.
Okay, thanks very much.
Yeah.
We have a Q&A here. So from April, so great talk, Tyler. I have a question about the Trop B, Trop C pair.
Yeah.
How widely distributed are those two genes? And do you find them in all tropolone producing organisms, or are they exclusive to fungi?
So it seems that although there's been limited research on all of the natural products from this pathway, and it's sort of really hit in recent years, there's been a big interest. But it seems that these are conserved in all of the cases where you have the stipidaldehyde core for the main core. Let me see if I can go back to that slide, actually, kind of give you a sense of yeah, so in all cases where you have this sort of stipidaldehyde as the functional gamma hydroxy tropolone core, we see that those genes are conserved and are in place. As far as I forget what the first question was. Could you repeat the?
So how widely distributed are the genes?
Throughout fungi or throughout just the?
I think both. I think so. The first part of the question is how widely distributed, and the second part is are they found in all tropolone producing organisms or just the fungi?
Yeah, so not all tropolones are made through this route. There are just this particular class. There are other tropolone biosynthetic routes that have been identified that don't go through this. I think primarily PKS related in some cases, but for the most part, this is what is conserved for this particular scaffold. As far as in other organisms, I believe that there are similar, sequence-wise, there are similar non-heme iron enzymes in other organisms, but they are not necessarily associated with tropolone biosynthesis. So I think this is mainly a fungal thing.
So maybe on this note, Tyler, I wanted to follow up about the substrate specificity of these enzymes. So
yeah.
How narrow that substrate specificity is, and have you probed other substrate? Because benzoic hydroxylation is obviously very attractive synthetically.
Yes. So we haven't published on that, but we have looked at some other substrates which can be dearomatized through the same dearomatization pathway. My former colleague Summer Baker-Dockery probed a lot of substrates for the flavin-dependent monooxygenase dearomatization, and so we were able to use some of those substrates as substrates for the non-heme iron ring expansion. There is a bit of limitation. This enzyme doesn't like a lot of substitution. It's pretty strict in what it will accept as a substrate, but you can get away with a little bit of changes to that core.